And we are back here with a stellar guest, Dr. Siddhartha Mukherjee. I know that everybody Googles everybody these days shamelessly. Somebody calls somebody, you start Googling at the other end. So all of you know about him. So I will not spend more time reading up his biodata except to say he is, he is somebody who can multitask more than I have seen anybody multitask. He's a champion doctor. He is a Pulitzer-winning author. He is a scientist, but most importantly, he's a deeply reflective person who is willing to push the boundaries and who's willing to question his own science all the time. So the best scientist is the one who questions their own science. There are other scientists who think that anything that I've said once will always remain the same, but the beauty of science is it always changes. So Siddharth, thank you very much for being here. And Kiran, I don't have to introduce her to you. If I have to introduce Kiran anywhere in India, but most of all in Bangalore, then I think I'll be accused of wasting time and worse. So Kiran, as I said to you earlier, she'll ask the intelligent questions. I will ask the dumb questions. The first one I'll ask, I've been tutored to ask this, so it might sound like an intelligent question, but that's because somebody told me to ask this. We can all talk about cancer, and we've all read your first book, and everybody's scared of cancer. So let me start with what's new. What's new right now is a cell in place of a pill. Because you say that what if you can cure a disease by using, cure the disease by using the cell instead of the pill. Will you explain that a little?
Sure. Well, first of all, thank you for having me. Thanks to Kiran as well. We are undergoing in cancer therapeutics, we'll talk about cancer prevention and early detection separately, but in cancer treatment, we're undergoing a series of very profound revolutions. As you very well know, for the longest time, the model for cancer treatment was based on cell-killing drugs, standard chemotherapies, and this was built upon surgery and radiation. So you really had three choices: surgical removal of the tumor, radiation to kill the cells, and chemotherapy, traditional classical chemotherapy, cell-killing chemotherapy to kill the cells. That paradigm began to evolve in the 1990s as our community of cancer scientists began to discover what the root causes of cancer are. What is the root cause of cancer? And we now know that the root cause of cancer, of course, is mutations or alterations in DNA, alterations in genes, and genes usually that control the division of cells, but can also control other factors such as how cells acquire nutrients in order to divide and so forth. So once those were found, there was an attempt to find new medicines, a new class of medicines, which would strike at the Achilles heels of cancer. So these were also chemical therapies, but they were called targeted therapies. And they included not just molecules like imatinib or Gleevec, but also antibodies such as Herceptin or trastuzumab. Finally, much later, as these paradigms began to evolve, we also began to understand that cancers are surveyed by the immune system, by our own immune systems. And often we began to understand that cancers grow in the presence of an immune system, often by evading immunosurveillance. And so the thought, this was an old idea, but had been not been possible to really incorporate into the cancer pharmacopoeia, was what if we could use these immune cells or other cells just as we use drugs to cure cancer? What if we could weaponize a cell, an immune cell, and by weaponize I mean add to that cell a virus that would educate that cell to go and kill a cancer and thereby kill the cancer cells. And this whole idea, which was in its infancy in the early 2000s, began to acquire a lot of potential, and now has become one of the most interesting arenas of cancer research. Let me be clear, not all cancers have been amenable to this kind of therapy. So cellular therapy is really a very broad term, but it uses this idea that you can use cells rather than pills or small molecules or drugs as if they were drugs. And it could be, of course, against cancer, you can use the immune system, but it also incorporates aspects that are not related to cancer. So, for instance, one part of my laboratory, we actually discovered, really by accident, I should say, we discovered the stem cells that form cartilage. They were not known before, cartilage and bone. So, one aspect of cellular therapy is regenerative. So, could you replace cartilage and very common disease across the world, particularly disease of women with an enormous burden is osteoarthritis. So we have a big now study going on in trying to use these cells, again as pills. We've been trying to use pills or drugs to ameliorate osteoarthritis now for decades and really not gotten very far. This would be the first time that one uses cellular therapies for regenerating, for instance, knees that have degenerated. You can use cellular therapies to alter, for instance, you can combine cellular therapies and gene therapies to cure diseases such as sickle cell anemia, beta thalassemia, another disease with a huge burden in places like India. So it is a very broad term, but it asks a very simple question, which is that the cell is a living, obviously a living thing. Can we use that, can we manipulate that living thing in such a way that it becomes a therapeutic entity? And if you can use that, can you use that in the context of not only cancer, but also other degenerative diseases?
So, before we go to Kiran, which cancers does this work on as yet, and which cancers, important cancers, it doesn't work on?
So the first cancers that cell therapies were used for were blood cancers, leukemias and lymphomas. The very first successful trials were all in blood cancers. I just recently profiled for the New Yorker probably I think she was the 7th patient, an 8-year-old girl named Emily Whitehead, who had multiply relapsed with blood cancer and was almost on the verge of dying, but she is now 15, as far as we know cured, is being monitored very closely, but as far as we know cured. So it works for blood cancers. More recently, it has worked for cancers such as lymphomas and myeloma, another cancer with quite a large disease burden in places like India. Where it's not worked effectively thus far is in the so-called common solid cancers. These would include pancreatic cancer, these would include ovarian cancer and many others, lung cancer. In these cancers, they have not been so effective, these cellular therapies. And one of the most interesting arenas of investigation is why. Why is it that blood cancers are responsive, and why is it that these solid tumors are not responsive? And we and others have actually spent some time thinking about this question and trying to rework our ideas and thinking, maybe we should try different cells, maybe we should try different combination of modalities. And there's all sorts of ideas about how to get these cell therapies to work in solid tumors, solid cancers, lung cancer, cancers of the pancreas, liver, and so forth.
Kiran, you have to play a double role because you are also a science person by yourself. Is this all foreign to us in India, or is there work going on in some of these fields in India as well?
Well, I think there is a lot of work going on in this whole area of immuno-oncology and cell biology, but I would say that we need to really catch up, because the reason why Sid and I decided that we needed to do this and needed to basically leverage science and technology and take it to the next level.
Needed to do what, if you can just explain?
Well, needed to start a company like what we are doing in India,
Immuneel,
is because it was very apparent to us that both US and China were the ones who were being talked about in this whole area of cell therapy. And as you just heard from Siddharth, I think the fact that you have this wonderful opportunity to look at cancer therapy so differently, I just felt we can't be left out of it. And with that in mind, knowing that we have such a huge cancer burden in this country, we had to do something about it, because these therapies, what Sid didn't talk about was the present cost of actually delivering these therapies in the Western world. The cost is absolutely unaffordable in a
Like
country
what?
like India. It can cost anywhere between $300,000 to $1 million per patient. And that's not something even the West can afford comfortably. So I think it was very apparent to us when you have these kind of, almost miracle therapies. You just heard about this young girl who is almost cured today. And the fact that these were death sentences up until now, and the fact that you can actually then pull them out of that death trap and give them another life, I think it is just amazing what's happening. And I think it's important that for a country like India, we have the scientists, we have the science, we have the clinicians, we have the whole ecosystem that can actually deliver on these therapies. So it was important for someone to really make something of this. And with Sid's help, I said, well, if you're willing to help, I'm willing to basically, partner you in delivering this therapy to India well, not by Indian standards at an affordable cost base, because, nothing is going to be considered affordable in India. But certainly we want to bring down those kind of prices I just talked about to a tenth of what it costs in the West. So I think that's a huge task ahead for us. But I think we are very excited, because what we did see is that we do have the capabilities. There are lots of small groups around the country doing work in this particular cell therapy. It's for us to now start populating the country with more such groups, and we will be one very important group who's going to be really focused on taking that living cell, that living drug into patients. So that's what we are
But I do
focused
hope
on.
this research in India is better regulated than, say, stem cell research, because lots of freelance arrangements came up there and people started offering stem cell therapy just because there was no regulation.
No, absolutely. I think regulations have to be very, very well constructed, formulated, and delivered, because I think, as you very rightly said, this is about something about delivering an altered cell back into the body, and it is delivered through a lentivirus. So you have to make very sure that that virus doesn't go off target and starts replicating. So there's a lot of quality control. It has to be done under the strictest of GMP conditions, because you don't want
What is
infection.
GMP?
Good manufacturing practice,
Okay.
because you don't want any contamination to be carried into the patient, because you're trying to cure a patient, not infect the patient with other things. So I think it's a very complex therapy. It's a very challenging therapy, but then the outcome is so compelling that we have to find a way of delivering it to patients.
You were saying something?
Well, I was saying, there was this, I just wanted to convey to an audience how exciting it is. Because you forget we're using technical terms here and there. There's a famous Asimov quote which says that the best science is sometimes almost indistinguishable from magic. There is a quality about, just think about this for a second, of the status of medicine in which we extract from the body living cells, weaponize them we infect those cells with a virus, we grow those living cells in an incubator so that they expand several hundredfold, and then we recollect those living cells that we have now genetically manipulated and put them back into the human body and use that as a drug. Just imagine the excitement. Imagine it should really send chills up your spine compared it to most technologies.
So, say in terms of medical science, what breakthroughs do you compare this with? The discovery of insulin, the discovery of antibiotics, the discovery of bypass surgery?
Well, so certainly, and I just wrote a piece about this. What's interesting is that these therapies, uh, the, what they're most comparable with is bone marrow transplantation. When bone marrow transplantation first appeared in the 1970s and 1980s, it was also considered something completely magical, or kidney transplantation.
Right.
These therapies sit in between what is the procedural and the pharmaceutical. They are procedures that are pharmaceutics. And that's why it's very important to concentrate on the procedure. In fact, we ourselves, unlike the rogue stem cell clinics, we ourselves have gone to the regulators and said, regulate us. We ourselves are going to the regulators and say, inspect us, because we want the highest quality of, we want the very highest quality of product. And there's a danger of this being deregulated. And so, given those circumstances, we've ourselves gone to every regulatory body and met them and said, look, this is the plan.
Because this is not something that you're selling off the shelf. This is not a brand.
This is personalized
Yes,
medicine.
this is not a brand that a doctor is writing and somebody dispenses.
Far from it, yeah.
Far from it. So for each one, you have to make this drug. So you are in the American environment, which also which allows great science, but also allows great superstition. Now we read about this great anti-vaxxers business going on in America. People are dying of measles now. Measles are back. So how do you sustain this in such a suspicious environment?
Well, look, in general, there have been so many conspiracy theories and nonsensical theories around cancer that you always have to battle, as it were, the superstitions around cancer treatment in general. Oh, it's a conspiracy, this, etc., etc. But there has never been in the United States, or for the matter elsewhere in the world, a lack of innovation. And it takes time. You have to. This is an active process. It takes time to convince people that this can be delivered safely. I was telling Kiran recently. Many of you might know this. In the course of the development of bone marrow transplantation, which by now has cured several blood diseases, and including several leukemias, when the first tests were done, 88 patients died. It was the 89th patient who lived and then began to show long-term remissions. So if you're a doctor or if you're the public, you have to have trust in the process of science such that you understand that these patients died either because they had a terminal illness or because the treatment was not successful. For what reason? What was the reason? Because everything arises in medicine, everything arises as a consequence of the project of reason. You have to apply reason to why you fail. You have to apply reason to why you succeed. You have to apply reason to when a patient gets a response but then relapses. All of these are, if they're done systematically, build trust. And part of the problem, I think, to be honest, with the anti-vax movement is that we as a medical community had become very complacent about the success of vaccines. What we should have been doing is telling people every year, year after year, is that the reason your child is not, having horrible diseases such as mumps, measles, and rubella, among other diseases, polio, is because we invented, as humans, we invented mechanisms to prevent your child from having these diseases. Instead, it became very routine. You went to your pediatrician's office, your pediatrician gave you an injection and said, now go home, right? But again, I have to convince all of you, the excitement that pervaded the world when the first polio vaccine was successful was just enormous. Children in the summers in the United States, of course here, when polio would sweep through the country, they couldn't leave their houses for the entire vacation. They would be locked up in their houses. In Boston City Hospital, there was a ward in which there was basically a line of so-called ventilators, because in those days we didn't have positive ventilators. They were negative ventilators. These were steel chambers which the children were put into, and the steel chambers would expand and contract because they had paralyzed the upper parts of their body during the acute phase of the polio epidemic. Imagine a row, 20, 30 such incubators with 10-year-old, 5-year-old children sitting with the steel pieces moving so that they could breathe. And now imagine 10 years, 15 years later after the introduction of the vaccine, those incubators, you can't even find them. You can't even find a picture of them. So the idea around the entire anti-vax movement was that there was the nihilism was because I think we failed to convey the excitement and the number of lives saved in the process.
I know. So I'm of the generation that still carries the marks of smallpox vaccines. In India now we have the 4th generation being born
Yeah.
without the marks of the smallpox vaccine.
Yeah, it's a miracle. I mean, as I said, these are incredible inventions. These are inventions that have left their mark on human history, and to be complacent about them, and worse still, to not give the scientists who put their blood, sweat, and tears into this is really doing a disservice to human innovation. We were talking today actually, and I said, we were talking today about how to accelerate the pace of what we were doing, and I just said, in science there is no 5-day week, and in medicine there is no 5-day week, right? In medicine there's always been a 7-day week. We have invented a 5-day week because we want to take 2 days.
As in journalism, at least we pretended to be that way.
Exactly. So one question was, well, how can we accelerate it? I said, make 5 days into 7 days. That's already, you've already gained 2 days of work, and that is the fundamental driving spirit of innovation.
So Kiran, let me ask you a twin question. One, what do you have to say for these anti-vaxxers? Everybody knows who anti-vaxxers are. These are very powerful lobbies in America and very popular now, which are campaigning against vaccines. That's why measles are coming back now in large parts of America. So what do you have to say to these people who fear science? And second, science, he said, is 24/7, 7 days a week, and all that. But how do you then combine entrepreneurship and science the right way? Because the moment that happens, then again, at the popular level, people begin to get suspicious that there's a profit motive. Oh, corporates have got in, right? So everybody starts getting into that Monsanto mindset. So how do you deal with this?
So first and foremost, I think like Siddharth just said, I think you need to connect with society. I think science needs to connect with society. I think scientists need to basically use simple language to communicate the marvels of science to society. And I think, there are umpteen examples of presenting to society why certain things are very compelling. I mean, I think, we haven't done a good job about communicating many things. I think we go through waves of fabulous innovations in science, in medicine, and then we start taking it for granted. This particular cell therapy, I mean, today we are at the cutting edge, right, in the early stages of this particular cell therapy. I mean, in the years ahead, I'm sure this will become standard of care. But I think every new innovation, whether it's a vaccine, whether it's bone marrow transplant, whether it's a new drug, I mean, if you look at the hypertensive drugs, you look at many, many new drugs that have been, brought about paradigm changes in the way we treat diseases, I think have, been very well appreciated in the early entry times. But then later on,
they become part of life.
We've taken it for granted,
yes.
and then, people then start sort of becoming complacent, and, you have these kind of anti-vaxxers and things like that. So I think it's about communication. It's about celebrating the miracles of vaccination, which we haven't done properly. I mean, even if you look at the HPV vaccines today, which
this is a very important story.
Being opposed in India also now.
Which can do so much. And yet you have people, sort of creating all kinds of controversies around it.
HPV vaccines can protect women, young women from cervical cancer for life.
And especially in a country like
like
India.
India.
Well, I mean, the number of the incidence of cervical cancer in India is enormous.
Enormous, yes.
And as you know, there was in not only were the vaccinators, people who were advancing the vaccine, often thrown out of places. There were a couple of people who were killed. Was it not?
Yeah.
Well, in
In-
India killed,
not
I
in-
don't
not-
know.
not in India.
Not in India, but
Beaten up over
beaten
there.
up.
But there is a great deal of now kind of Swadeshi opposition also.
Yeah, I'm not talking about India. I was talking about
that
other,
this is
other
some
countries.
kind of a conspiracy to bring in one more sort of corporate product into India. There is so what you say about communication is great, and that's why Siddharth, you are so valuable because you are now making this. You are idiot proofing it for people like us. Otherwise, and these days every patient is a doctor. People go to a doctor. Doctor doesn't touch the patient. The doctor doesn't poke you, nothing. Doctor looks at the scans, prescribes something. By the time he or she is reading the prescription, patient is Googling side effects, right? That's what happens. So it's very important that we hear about cutting edge research in a manner that we can understand.
And let me just add one more thing. In this particular case, you're talking about terminally ill patients.
Right.
They have no hope left. This is giving them the greatest hope, and you're seeing these wonderful, wonderful outcomes in many, many patients. And another thing I also want to tell you is, like everything, there is a risk. Nothing is risk-free, and I want people to understand that.
Yeah.
Because just as Siddharth said, when you started bone marrow transplants, it was the 89th bone marrow transplant that was successful. And we must understand that even in simple things like vaccinations, some people react very, very adversely.
Penicillin can still be fatal.
Absolutely.
I mean,
so I think we have
I have to
to
qualify,
understand.
can be fatal in the rarest of rare cases. Otherwise, somebody will take out 5 seconds. Shekhar Gupta said,
Penicillin
can be fatal.
Well, my aunt died of a penicillin anaphylaxis. But I think what is important is any new therapy is inherently risky. And therefore, we want to do it very carefully, in a very informed way, and we must protect ourselves in every possible way in terms of making sure that it is as safe as it can be and as efficacious as it can be. So that's why I think it is important for us to remember that this is not a simple drug. This is a complex therapy.
In fact, the birth of cellular therapy in the United States and in Europe was also marked by many deaths. The FDA had to shut down certain trials and had to examine them. The important thing is of course, the legacy of science is a legacy of failure. And that legacy, the only reason science works is by examining that failure instead of incriminating it. And that's very important, because if you examine the failures rather than incriminating it, of course there are frauds and there are examples in which the appropriate measures were not taken. But when the appropriate risk mitigating measures have been put in place and there's a failure, there is the center, the core of innovation is to examine failure rather than incriminate it. And if you do the opposite, if the culture turns into one of incrimination, then all innovation dies. What innovation, which culture of innovation has been a risk-free culture? Risk is the center of innovation. Mitigated risk is the job of scientists and doctors. We spend hours and hours and hours of our time thinking before acting. And this is part of the reason that complex therapies were born.
So, Kiran, the second part of that question, you then marry entrepreneurship with research. Because ultimately, everything has to scale up. To scale up, it has to build an economics. The moment you do that, you run into this wall of suspicion. And then you come into public health care and private health care and who can afford what. Equity in healthcare has now become a global issue. I think in America it's going to be a big issue in this election.
An enormous issue, yes.
Enormous issue going ahead. So how do you deal with that?
So as I said, for India, the model had to be affordable access as much as we could do. But what is interesting about Immuneel is we've created for the first time in the country social capital. We've created philanthropic, a combination of philanthropic capital and risk capital to actually deliver this very important
How have you done
therapy.
it?
So the investors in Immuneel have been philanthropists, have also been VCs who have given risk capital to us. And I think it's a very, very interesting model because remember, if a pharmaceutical company had to actually go about starting and developing this particular cell therapy to take it to patients, I think the upfront investment costs were going to be so huge that I don't think shareholders would have been comfortable with taking that kind of risk. If you looked at even a hospital trying to do it as a service, they can't because this is too complex a therapy.
Right.
So therefore, it had to be done through creating a fund which was going to be able to do everything we could in the shortest possible time. You don't want to take too much time because you had to catch up.
So part of
Others
that
were
fund, part
doing
of
so
that
much
fund is
more.
philanthropic contributions.
Yes. So we have raised the initial startup capital. It is a startup. I mean, for the first time, again, after many, many years, I've started my next startup after 40 years. So it's a very exciting process for me as well. But the interesting thing is, this is a very interesting startup because it's about accelerating the therapy from the cell to the patient. And I think that is very exciting because we've got a bunch of scientists and others who want to really be a part of this journey. And I think the investors also are very excited about being part of this very exciting journey because this will put India on the map of cell therapy. If we hadn't done this, we would have just had a few groups tinkering around here and there, but you would not have got this cell therapy get to patients and make that difference.
You need scale.
And I think that's very, very important. So we are people, I mean, I at least know, I've had this experience, I've basically know what it takes to commercialize such therapy, not this particular therapy, but certainly I've taken risks. I know what it takes to take this to patients. And I think therefore, everyone who's engaged with this mission is on a very, very noble mission of bringing these cell therapies to patients in India.
Well, Kiran's just given me the cue to do a little bit of self-promotion. Just as she says she's doing a startup after 40 years now, another startup. I've set up a startup after being a journalist for 40 years. So people do check out ThePrint and do subscribe and do read. And like she says, she's trying to build an economics. I'm also trying to build an economics around fair journalism in India, fair and correct journalism in India, with the belief that there is no contradiction between good journalism and the marketplace. Having said that, now we have to come to questions right there.
Hi, sir. Sir Furkan here. Hi, ma'am. How are you?
Please introduce yourself.
Furkan. My name is Furkan.
फुरकान खड़े होके बात करो ना ताकि कैमरे में आप नजर आओगे. हाँ. इतनी अच्छी टी-शर्ट पहनी है आपने. हाँ.
So basically, sir, I wanted to ask more to Kiran, ma'am. So, ma'am, recently that book came about the generic drugs, the issues with the generic drug, the bottle of lies. So, what has gone wrong in the Indian pharma industry? What do you think? Because only company that was not that came clean in that book was yours, and most of the other companies were named in that book. So, what has gone wrong, and what can you elaborate more on that, ma'am?
Well, I don't think I really want to comment on that book because it talks about an incident that happened more than 10 years ago with Ranbaxy, and then much of what has been written after that is very anecdotal. It is an unfair bit of journalism. I guess it's sensationalism. It sells copies, but I really don't think I want to comment too much on that. But I think what that book tells you is how important it is to make sure that you follow the right processes, and especially for a therapy like this, it's very, very important that there are no shortcuts taken.
Thank you so much, ma'am.
Question right here, microphone. Did you want to add something?
No.
Microphone right there.
Dr. Jagdish Shinnappa, I'm a practicing pediatrician for the last 40 years, and much of what you have said is
Set up a
resonates.
startup, 40 years.
Yeah. Yeah. Actually, I've actually handled polio babies with iron lungs for a long time when I was a student. Well, as Shekhar mentioned, Siddharth mentioned that it's not seen any longer. I had only one question which I wanted to ask both of them. What would be the ethical challenges in introducing a therapy like this, the West compared to our country? Because in our country, if you look at the HPV trial which was done, the basic problem was the ethical challenges. Because it was a challenge which was done with very bad informed consent and in a vulnerable population. So I think those are going to be challenges when you are going to introduce a new therapy like this.
Well, you know, the templates for informed consent are already very complex. They're especially complex in pediatric cancers, because usually the child doesn't give consent, it's the parents who are giving consent. The capacity of any of this going wrong in any phase, for instance, being contaminated or not having a QC release, not being able to release it because of quality control. So you give the cells, we process them, but at the end of it, nothing comes out because it doesn't pass the quality control. This is a complex idea to convey to the parents of a child, let alone the child himself or herself. How much hope do you give to a child with acute lymphoblastic leukemia who's going to have this therapy? How do you explain the fact that maybe in this particular case they did not reach a remission? What if they relapse afterwards? So the ethical challenges are very much the challenges of medicine, the standard challenges of medicine. Appropriate consent, not using this on populations that don't understand the complexity of the challenge, and yet on the other hand, making it accessible. You don't want to create a barrier to access just because you can't explain the complexity of the entire system. They are quite large. They're actually not specific to India. They're quite wide. We will use the best available practices. Consenting, as you know, consenting a child with acute lymphoblastic leukemia who's 2 years old to what would amount to 2 full years of chemotherapy, including potentially therapy that's injected into your spine, is not easy either. How does that child understand that they're going to be, that for 2 years they're going to be confined, basically confined to an IV pole, and with the possibility of getting painful injections into their spine? But those challenges are universal. We will try our very best to try to address these ethical challenges. Some of the challenges are very unique to cell therapies. These include the possibility, which has been noticed, that sometimes these cell therapies are so active that they create their own problems. Not only are they good drugs, they are too good drugs. In fact, one of the reasons that the FDA halted one series of trials, one of the series of trials, is that patients were dying not because the drugs were ineffective. They were dying because the drugs were so effective at killing the cancer that as the cancers were melting away in these children and adults, the body couldn't handle the consequences of the immune attack on cancer. The body imagined as if this was an immune attack on itself, because of course the cancer is part of the body. So all of these challenges have to be handled. The appropriate consent forms and the appropriate regulatory forms have to be handled correctly.
And therefore also the trials that need to be conducted have to be very carefully conducted.
And in India, it's a big challenge now because of the suspicions of the past.
Yeah, but I think slowly, I think people realize what a disservice they've done to innovation by banning clinical trials to begin with. So I think the government is now beginning to revive this whole
but they are not reversed yet. The ban still remains.
Well, I think it's the way you interpret it. I think they do allow you to carry out clinical trials.
Right.
They're still very, very nervous about first-in-human trials. And then they have this strange logic that if it's a made-in-India drug, you can try it. But if it's a foreign drug, there's a lot of xenophobia about clinical trials. So I don't see.
But some of it is, of course, the history of the colonial experience. You don't want to, and in fact, in the United States, the history of using African Americans
Right.
as trial subjects without appropriate consent. The famous,
There is
there is,
mistrust. Yeah.
of course, there is a rich.
But it can go too far.
Absolutely, it can go too far. The appropriate balance is always the fine point for an innovator.
Before we take that question, let me ask you again an idiot-proofing question. You say that you take these cells from the body, you weaponize them, you make them nice guys, you give them
Nice guys, we weaponize them. Yes.
Yeah, you give them the sniper rifles, go and kill the bad guys. So first of all, before you insert them back into the body, what mechanisms do you have to see that to make sure that they have the right weapons, and two, that they have the right intentions? So can they go inside the body and change their mind, or can they become double agents, body filled with mataharis, right?
So as I said, I told you, I gave you one extraordinarily important component of this is the so-called release criteria. So this therapy.
Because once it's inside, you can't mess with it.
Well, you can.
You can.
There are many ways once they're inside to quieten them down. In fact, that is the basis. And unless you have someone with experience, you don't know how to silence these cells if they go rogue, as it were. But all of this said, the first step, the first question you asked is that there are very stringent criteria by which the cells are released. And this is also true, by the way, for stem cell therapies, and should be true for true stem cell therapies. Bone marrow transplantation is a stem cell therapy, and before implanting a bone marrow, you have to do 1,000 things to confirm that you have the right yield, that you have the right cells, that the cells have come from a right donor, that there will not be a mismatch, or et cetera, et cetera, et cetera. By analogy, by the way, the same thing is true for any transplantation. So by analogy, there are a whole set of release criteria, and if the release criteria are not met, then the product is not released. The second fact is that there are all sorts of medicines that are now available that allow us to dampen the immune response if it goes haywire, because these weaponized cells are, of course, can be quite dangerous. So the success of the study depends on, again, the use of medicine. And here is where we convert from the science of medicine to the art of medicine. The clinicians who give these therapies have to be trained in the art of medicine, which may be that you make a decision based on very fine-tuned ideas about, is the balance out of control? Is the immune system now, this weaponized immune system, is it acting up in the wrong direction? Should we dampen it? To what extent? When should we take off those dampeners again? This is the art of medicine, and this, of course, requires someone who's quite practiced in this.
And in fact, this is one of the key management criteria for the patient. It's called cytokine release syndrome, where your body and your immune system goes hyper. But that's why you need a good clinician to actually dampen it.
And the science of medicine and art of medicine is a fascinating point. We'll come back to that. Question there, sir? Yes, a little bit louder.
I don't think this is working, right?
Yeah,
it's now it is. Yeah, hi, Ravi Narayan. I run a software company. I head a software company here in India, so I'm a novice to most of the life sciences, but I have a question. And this is to, Kiran mentioned that there is risk in everything, and that is true. But as a discerning member of the public, I think we owe it to ourselves to assess and quantify risk in any decisions that we take. So to that end, I have a few questions about how do we quantify risk. So, for example, is this a therapy that's been FDA approved? And secondly, what is the hit rate or the success rate on patients? I heard one example, but are there numbers to quantify success rate as it is today? I guess it's in the US. It doesn't sound like it's been introduced into India yet. And to build on what Shekhar had mentioned earlier, how targeted is it? So we know that cancer cells, the current therapies on cancer cells are not very targeted. So radiation or chemotherapy definitely affects the good cells as well and leaves you pretty impoverished at the end, even if you succeed in fighting the cancer. So how targeted and how selective is this therapy? So 3 questions, I guess. Thank you.
The first point is that, yes, the FDA has approved now, over the last 2 years, the FDA approved 2 such drugs. This year, it is likely to approve 2 or 3 further such drugs. The success rate depends on the particular drug. So they range from, just to give you some range or some sense of what the range might be, in multiple myeloma. First of all, remember, almost always, you talked about risk mitigation. Virtually all these trials have been run on patients who are terminally ill. So their chances of death are virtually 100%. They approach between 90 to 100%. So to give you Emily Whitehead's case, in the absence of CAR T-cell therapy, her mortality rate was 100%. She had failed or gone through 2 attempted bone marrow transplants, and both of them had not worked. She was now in full-blown leukemic phase, so her mortality rate was 100%. Therefore, the risk mitigation becomes quite extreme, because you're facing the possibility of a mortality rate of 100% in an 8-year-old child versus whatever experimental. She was, of course, an experimental subject. So just to give you a sense of what those numbers look like in multiple myeloma, now with the new drug that will almost certainly be approved by the FDA, you take patients with, again, mortality rates in the 90–80%, and about 80% will achieve a remission, which is already a big step. And of that 80%, on the order of 50 or 60%, so give or take about 50% in total of people who had a 100% mortality rate are alive at 2 to 3 years. Now, this is a continuing process. 2 to 3 years is already quite a large period of time. So, and some of them have been alive now for 5–6 years. We saw this paradigm evolve when the first targeted therapies also came around. That over time we got more and more comfortable with them. The one key factor is you have to realize that cancer is first of all, is an evolutionary disease, in the sense it follows the laws of evolution and natural selection. It is like watching Darwin's Galapagos Island in a perverse form inside your body, because the cancer cells are constantly evolving. Your body is constantly evolving in response to it. So therefore, any therapy for cancer is really a mathematical game between the cancer and you. So when you apply any therapy in cancer, it really depends on the numbers. If you're trying to use the therapy to kill 1 billion cells, then it means that 1 billion cells have the opportunity to become resistant and therefore cause relapse. If you use the same therapy on 1,000 cells, then you have only 1,000 possibilities that the cells will become resistant and relapse. Finding the right point, as it were, between the 1,000 cells and the 1 billion cells, where the risk is exactly appropriate, where the risk becomes, because all therapies carry risk, where the risk becomes appropriately mitigated is, of course, an extremely important question. And the only way we can answer that question is using science, is using reason, not anecdotal evidence. And that's why we undergo clinical trials and try to find the so-called sweet spot in which you apply the therapy, which has toxic consequences and is expensive, to the right kind of patient. And we've done this over and over and over again in the history of cancer.
Kiran, you want to add something to this?
No, I just want to say that, look, I think, cancer is such a disease that you have to keep innovating. You have to keep trying new treatments. I think any cancer patient will know that the moment they are not responding to one drug, they want to try something else. So this is one disease that needs constant innovation. And I think we mustn't forget that. So I think when you keep on looking at that mathematical question of risk and you know, assure me that this is going to work and assure me that I'm not taking a risk. I don't think cancer patients ask that kind of question. I think cancer patients want hope. Cancer patients want to try any new experimental drug that will save them. I think that's what it's about.
Siddharth, you talked about this tussle between the body and the cancer cells, or the treatment and the cancer cells. Your new book is Gene. Do cells or genes also have, does a gene or a cell have a mind of its own?
No,
Or is it a bot?
it's not. It's neither a bot nor does it have a mind of its own. It's an, you know, these are obviously living elements.
So it doesn't change its mind, it just mutates physically.
That's correct. I mean, the mind is its own unique thing, but cells are living things, and as living things, they are malleable. They have the capacity. The most important capacity is that they have adaptability and evolutionary adaptability. By adaptability, it means that they can change their internal software, which is and once they change their internal software, they can use that change in the internal software to live in different environments, to do different things. That's the change in the software. And if you want to continue that analogy, they can also change their internal hardware, which would be their genes, the DNA, and thereby become altered. So a cell could be imagined. I mean, you know, this was, of course, a very insightful idea that was brought into biology from physics. A cell is really a collated version of information. You can think of cells in many different ways, but a cell is an information processing machine. It is a living machine, but is an information processing machine. It takes in information from the environment and it puts out information to the environment. Now, as I said, there's a software component to it. The cell can readapt the software, internal software, and change itself so that it can now operate in different ways. And in certain circumstances, it can change its internal hardware, that would be genes, mutate that hardware, and then acquire different characteristics.
Question there. Sir, who has a microphone here?
Hi, my name is Bhairavi, and first, thank you for a very fascinating.
Tell us your name again, and what do you do? Tell us.
Bhairavi.
And what do you do?
So I'm an entrepreneur in the field of healthcare, and I think this is a very fascinating and important discussion that we need to have. But I'd like to come back to a thing that you mentioned earlier, which is, you know, the importance of effective communication. And if you consider the fact that in India we see many more cases of advanced disease as compared to, you know, early stage illness, and also if you consider the continuum of cancer care, the problem really begins at the beginning, which is, you know, prevention and screening. So I just wondered what your thoughts were about, you know, effective strategies for overcoming sort of the stigma associated with screening for cancer, and how we can address that problem in an effective way in both the urban and rural populations.
Yeah, I mean, to be very, very clear, if you wanted to have public health impact in virtually every disease, the public health impact has occurred from prevention and not from other modalities. So the primary effort of seeing a public health response in cancer will come from cancer prevention. It's really a pyramid, and one should imagine the pyramid carefully and think about the pyramid carefully. The bottom of the pyramid is prevention, cancer prevention, and over the years, usually that task has been taken over by governments. That is the task that is best assigned to governments because they have the capacity to change the social milieu, as it were, such that you can advance cancer prevention campaigns. Our government has done, the Indian government has done quite a poor job, for instance, in tobacco control, which would be a simple mechanism of cancer prevention. We gave a couple of other examples. Human papillomavirus vaccination would be an example in which you could potentially eradicate cervical cancer, or a tobacco control program would reduce the risk of lung cancer by factors of 10-fold or 20-fold, as we've seen in the United States and other places. So that is the base of the pyramid. The second layer of the pyramid is early detection, as you're talking about, finding cancer in its early phases. And there usually there is I would say, a public and private partnership that works effectively. the problem with cancer prevention, early detection, is that our mechanisms of early detection of cancer are still in their infancy. So aside from a few cancers which we can name, breast cancer through mammography, colon cancer through colorectal cancers through colonoscopy and other screening methods, and 2 or 3 others, we really don't have very effective cancer screening mechanisms, early cancer detection mechanisms. And that's a problem because, as I said, if it's a numbers game, then the earlier you detect the cancer and apply the therapy, whatever the therapy might be, then of course the better the outcomes are. So here is this is an area that is rich, extremely rich for investigation, and there have been a number of groups that have deepened their investigation into how to do this prevention and early detection. In fact, our group itself has changed. One part of our group has focused now on metabolism and diet, which we had ignored before. We had a big paper out in Nature, which is widely covered, in which we use diet and chemotherapy in combination for cancer therapeutics. The final phase of the pyramid, as it were, is cancer treatment. And if you look at cell therapy, that's the pinnacle of that final phase because of the complexity of the treatment. But you have to remember that all of this is part of the ecosystem of cancer. You learn about cancer prevention from cancer treatment. You learn about cancer treatment from cancer prevention. This all forms a kind of ecosystem, and when it's successful, you can begin to see public health impact on cancer. You know, not to the satisfaction of everybody. This is a disease that we all dread, but certainly in places like the United States, cancer-specific mortality has decreased by about 1 to 2 to 3% every year, and has been decreasing since, I would say, since the 1950s, and that's been a real success story. You might say 1 to 2% a year is not that great, but cumulated across 20, 30 years, you get a 20, 30% decrease in certain cancers. Some cancers have been very resistant to being amenable to therapy, pancreatic cancer being one of them, gallbladder cancers being one of them, liver cancers and so forth have been very resistant. Others, such as breast cancer, transformation. We are now seeing 30-year, 40-year survivors of breast cancer. We are seeing 10-year survivors of metastatic breast cancer that's spread outside the breast. We are seeing 10-year survivors of metastatic colorectal cancer that's spread outside the colon or the rectum. So in some cancers, the effect has been transformative. We are seeing 40-year survivors, 50-year survivors of childhood leukemia. It really varies, but the effect has been present and there in most cancers.
Hi, my name is Sujata. I'm a trained immunologist and I haven't practiced in the last 12 years, but I've just written a book on preventive health. And my question is about Ayurveda. Since you're doing this trial in India, and I'm sure it's not part of the plan right now, but is there a plan to use Ayurvedic medicine as adjunct therapy to support the side effects that can be expected?
I don't have such a plan. I don't know if you have such a plan. I mean, right now we're in the infancy where we're making lentivirus in sterile containers and infusing them into T cells. How we ultimately deploy these therapies and use adjunct therapies to palliate and to palliate symptoms and for symptom relief is certainly a downstream consideration. But right now we're in the thickets of the woods, as they say. We're in the thickets of the woods. Our conversations are around, you know, how many particles of lentivirus are you going to use per cell? Where is this cell going to be grown, and what are the quality control release factors that will allow us, you know, should we do flow cytometry or qPCR to figure out whether the virus has gotten into the cell or not? So your questions are very relevant to patients, but right now we're, as it were, we're in the middle of the thickets of the woods, and your questions will become relevant once we have the first patients that we've given therapy to.
So Kiran, let me ask you a question, and the other side of the same question to you. What do you find, if you had a choice, would you be an entrepreneur or would you be a researcher in the same field?
Well, I think I'm a born entrepreneur and I'll die an entrepreneur. So I don't think I can answer that question
and
any
you've given
other
a
way.
chance,
But
would you-
I would say that I'm an entrepreneur who has been excited about research, who is driven by research, and who invests in research.
And who invests sometimes in news media startups also. Go ahead.
Do I have to choose between being an entrepreneur and a researcher? Well, look,
You can say, you can tick both.
well, I sort of have to tick both. One of the life cycles, I have to say that for the particular person that, or the particular niche that I inhabit is a so-called translational researcher. This was an idea dreamt of in the 1950s, that someone would have to take all these astonishing ideas that were emerging from the science of biology and apply them and make human medicines. So there would be a translator between one and the other, and if you imagine medicine as a relay race, the baton would be passed from basic science to people like me, and then would be passed on from me to clinicians who would then deploy the medicines into humans. There is no greater joy that one can experience as a translational researcher than to see a medicine that you have dreamt up one morning, that you made a diagram of one morning. You woke up one morning and you wrote down on a sheet of paper, this is going to be a medicine, a human medicine. And to watch that human medicine come alive and treat a human being, there is no greater joy, and I would say, I would make the argument there is no greater joy that anyone can experience, period. You cannot experience this joy unless you've done it yourself. You have taken, you have applied, you've taken the benefits of reason, the great revolution that was handed to us from the Renaissance. All the way, you've taken that idea, put it down on a piece of paper, drawn a diagram, brought it to a laboratory and made a human medicine out of it and watched a human being being cured of a lethal or a non-lethal disease. I would say there is no greater joy. You cannot feel the shivers with any other form of activity. There's nothing else that compares. It is like the highest, it is like a kind of, it is like feeling a kind of, it is the highest high that you can feel. And you cannot imagine, I can't even convey to you, as we make medicines, I've now made several are in trials, several are in, I cannot convey to you the quality of the joy.
But we can feel it. We can feel it in your voice.
Yes.
Now, I know that we are running out of time, but I can't not have a question from Sandhya Ramesh. Sandhya is our science editor. We have a very strong and very extensive science section. She runs it from Bangalore.
Yes. My question is in terms of both the current political climate in the US as well as for the cancer research community. What is the scientific consensus and also your personal opinion on the use of marijuana and CBD to work with the side effects of cancer treatment?
Well, as you know, I mean, you know, the US is going through some important internal reflection about the use of both marijuana and CBD. CBD has to be evaluated properly. The effectiveness of CBD has to be evaluated properly. The use of marijuana in palliative care has been established, but whether it should be, you know, how to regulate it, how appropriately to regulate it, has been a big concern. As you very well know, recently there were 450 odd cases in which the vaping of whether it was marijuana or marijuana laced products or other products have caused lung diseases. So every time you open up the channels for the use of a substance like marijuana, you have to pay very careful consequences. And the CDC has been following this. The Center for Disease Control has been following this very carefully. So we'll see how it turns out. CBD is under evaluation, so we don't know yet.
So, do you have any closing comments before we go?
Well, I just think that this is a very exciting phase for all of us at Immuneel. Secondly, I think in cancer therapy itself, I think we are at the cutting edge, and it's great to be, you know, bringing that to India. And I think we need to really make sure that people support this kind of research both in the laboratories and in industry, and I think that's extremely important.
And you are still hoping that clinical trials, the rules relax a bit more.
Well, I'm very confident that it will relax.
It has to happen. So thank you very much. It's been a great conversation. I know this can go on, but this is Bangalore. You also need 3 hours to reach home. So we'll let you go. But you know, just a thought as you go away, just think about all the treatments that we take for granted today. And just think then, or Google, when was the first time such a thing happened? I can tell you a simple thing like a laparoscopic surgery on a torn ligament of the knee. It didn't happen until 35 years ago. The first heart bypass was just over 40 years ago.
Yep.
Just over 40 years ago. Anybody over 50 right now in India does not carry those 2 smallpox or 3 smallpox scars that my generation does. So when a breakthrough comes, it scales up really fast. And it scales up so fast that after a while you forget when it happened. And I'll give you a real life experience. I have a tough time, this is the anniversary of the 65 war, etc., etc. So history comes back, and now with this government, a lot of the history is being revised. So how did Lal Bahadur Shastri die? But if he had a heart attack, why couldn't they have done to him in Tashkent all the stuff that we do these days for heart attacks? To tell people that there was nothing for a heart attack in 1966. There was no bypass, there was no angio. I'm not even sure there were proper ECGs at that time, because we all now take it all for granted. So I think this is a new idea. I think very few people have it, but when it scales up, then the economies of scale will build up. And I think you might be looking at one more miracle, and I hope both of you are right, and wish you all success.
Thank you.
Thank you very much. We'll have Vivek from IIFL just come in for a quick felicitation and our team picture. Our team is just 5 of us here. It's okay.
Can we take these off?
See, I spared you the closing to the camera.
Can we take these off?
Yeah, sure.